So my name is Helen Chu. I'm at the University of Washington, and this is the next in our series of the Viruses and Vaccines seminar. And today I am really delighted to welcome back Trevor Bedford to give the SARS-CoV-2 genomics update at the beginning of the talk. And then this will be followed by a lecture from Professor Barney Graham from Morehouse School of Medicine. So Dr. Graham is professor of immunology, virology, and at Morehouse School of Medicine. His background is in research on respiratory viruses and vaccines. He has really contributed such significant advances to the development of vaccines for Ebola, for Zika, for RSV, and for SARS-CoV-2. So he did his MD at University of Kansas, did his residency, chief residency, and ID fellowship and his PhD at Vanderbilt University prior to joining the Vaccine Research Center at the NIH in 2000, from which he recently retired, I believe, in 2021. He has really contributed over 500 publications in the field of vaccines and structure-based vaccinology, and is best known for his research on RSV pathogenesis, structure-based vaccine design, and the application of mRNA delivery technology. He has received many, many, many honors in his lifetime. I can't list all of them, but he was named one of the world's 100 most influential individuals and one of the heroes of the year in 2021 by Time Magazine, and recognized as the Federal Employee of the Year by the Partnership for Public Service. So we're really delighted to have both Dr. Bedford and Dr. Graham here with us today to give us an update on SARS-CoV-2 as well as RSV. So thank you to both of you for coming. Oh, and questions in the chat, please. So I will go ahead and turn it over to Trevor to start.
Thank you, Helen. Yes, it's an absolute honor to be presenting briefly ahead of you, Dr. Graham. Really looking forward to your talk. So just going to take a few minutes to talk about SARS-CoV-2, and in light of kind of where we are now over 3 years out, trying to kind of give things a bit more context rather than just talking about the latest variant. So, so if we look broadly, I think this is maybe the most important graph that we could have at the moment, where worldwide deaths per day had been over 10,000 for a long while during the pandemic, kind of horrible numbers. And basically we have this divide in spring 2022. This peak here is the Omicron wave. And that was effectively like that was like almost an exit wave to the pandemic, where after this point, we can draw a line between kind of before widespread immunity and after widespread immunity through vaccination and through natural infection. And so now we've been having continued infections throughout the world. This peak here is the Chinese epidemic, where there wasn't kind of enough immunity. So you still see a peak there, but basically we still have lots of circulation, but infection fatality ratio is perhaps 10 times lower than it was before. And so this is still a number, 1,000 deaths per day, which is a lot, but tenfold lower than it was before. However, along with this immunity comes pressure on the virus to evolve. And so this is now sampled kind of throughout the pandemic. We have the initial Wuhan outbreak right in the center over here, and then the initial emergence of the variants that we all kind of know and love, Alpha, Beta, Gamma, Delta. Omicron emerges. And what's important here is if we look in the last just over a year, year and 5 months, that the amount of diversity here is now kind of rivaling what we saw before. And there are a number of very important labels that we'd attach things that I would think should have been kind of named Greek letters, but here we are. And so if we look at each of these, we can see continual emergence of a new variant and rapid displacement of what existed before. So initial Omicron rapidly displaces Delta, BA2 rapidly displaces BA1, BA5 displacing BA2, and on the most recent wave was XBB 1.5 that is basically dominant throughout the world at the moment. And this pattern in which you see kind of new things emerge and just so quickly displace other things is not seen. For flu, this takes years for a new variant to displace what was circulating. You can ascribe this to escape from population immunity just by looking at S1 mutations. So these are going to be mutations that will escape from antibody binding largely. And we can see that there's been, again, continual increase of S1 mutations even in this Omicron period. And if we just draw some kind of simple regressions of time versus S1 mutations, this is the entire pandemic. We get this pretty strong slope. We get basically the same slope within Omicron. So it's not that actually things have slowed down at this point. And if we compare that rate to kind of the relevant bits of other proteins from other viruses, we see it is quite fast. So you should be able to visually compare these slopes. So over twice as fast as H3N2, over 3 times as fast as H1N1, compared to like seasonal coronaviruses, maybe almost 10 times faster than seasonal coronaviruses. Exactly why SARS-CoV-2 is able to maintain this rate, I'm not fully sure. And so then in this light, we have the kind of the latest variant of XBB 1.6, but this is just the latest salvo in like this ongoing evolution of SARS-CoV-2. What's one thing to highlight here, however, is we get this interesting pattern where we have all of these S46P mutations. They occur again and again and again and again on top of this XBB background. And so this is kind of evidence that natural selection is really pushing for this 46P mutation. This was this big wave that we just talked about that is basically dominant globally. And XBB 1.16 has just a couple other spike mutations on top of this and a handful of other mutations elsewhere in the genome. But that's enough that right now we're seeing XBB.1.16, which first emerged in India, really taking off elsewhere. So we project that it will become dominant in June in the US, and we're seeing that takeoff elsewhere. And again, what's important here is the scale, where if we look at our estimated growth advantage of kind of between the variants, how many secondary infections do they cause? We see that XBB.1.16 compared to the current dominant variant of XBB.1.5 is perhaps 20 to 30% more advantageous. So only this like small handful of mutations seems to create a virus that is much more fit and much more able to spread. And so basically imagine this like flu, it continues to circulate every year, is able to cause lots of these reinfections based upon this continual antigenic drift. And I don't see a reason at the moment why this would dramatically slow down, suggesting that we should be kind of very on top of vaccine updates and all of these things. And that I will stop there. If you have any questions, I'll address them in the chat. Thank you.
Great, thank you, Trevor. In the interest of time, we're going to move on to Dr. Graham's lecture.
Good afternoon or good morning. And Trevor, thank you. That was a great overview. I'm going to be talking about another virus. I'm going to be talking about respiratory syncytial virus and how that whole field has evolved over time, and in part how that field informed some of the coronavirus activity. As you heard, I'm now at Morehouse School of Medicine in Atlanta, and I'm Senior Advisor for Global Health Equity, and very much focused on how to distribute vaccines fairly when they're finished. So we're going to end up talking about that at the end. These are my disclosures. I'm an inventor on vaccines and monoclonal antibodies for several viruses, and I'm on several advisory and boards and consulting for pharmaceutical companies. RSV is a virus that infects everyone by 2 to 3 years of age, and then recurrently throughout life, and causes a lot of infection and is the leading cause of hospitalization in the US in children under 5 years of age. Severe disease in infancy predicts wheezing and asthma in childhood, and sometimes long-term lung disease. And vaccine development has been complicated by a legacy of vaccine-enhanced disease that I'll review. Back in RSV was discovered in 1956 and then associated with bronchiolitis the next year by Dr. Channick, who ended up at NIH. And this was happening right at a time when polio vaccines were so successful. A whole inactivated polio vaccine from Salk was reducing rates of polio, and so a lot of vaccines in that era were made by isolating virus, killing the virus one way or the other, and injecting the virus and hoping for the right outcome. And this was done in RSV and in 4 large cohorts of children. In the youngest age cohort in children immunized before 6 months of age, 31 children were vaccinated. 20 of them were infected during that next winter season. And of those 20, 80% were hospitalized and 2 died. Usually hospitalization rates for children are more in the range of 2% of all children end up in the hospital in those first 6 months. So this was a vaccine-enhanced disease, and it really stopped vaccine development for a few decades until people could start studying this. And Brian Murphy, who took over the Channick lab at NIH, did this study with the sera from those children and found that in natural infection, children were making both a fusion inhibiting or neutralizing antibody response and a binding or ELISA-based type of serologic response before and after natural infection. But in vaccination, even though the binding antibody was reasonably robust, most children did not make very good neutralizing or fusion inhibiting antibodies. So there was a paradigm that a lot of binding antibody, a high binding to neutralization ratio, may be one of the problems. Indeed, a few years later, in Ruth Karen's lab, Fernando Pollack showed in those lungs from the children who died that there was evidence of immune complex deposition and complement fixation that did not exist in the pre-immune sera used to stain the lungs. And so the idea that there was a problem with the antibody response resulting in immune complex deposition because of so much binding antibody without enough neutralization potency was one cause of the severe airway disease. The other part was done in animal models primarily, and it was shown that priming animals with a whole inactivated virus, formalin inactivated and alum, led to an IL-4 response, dominant response in lungs post-challenge, whereas typically primary and secondary infection would be interferon gamma dominated response. And so this just adds to the obstructive disease that happens in children with severe RSV that comes from sloughed epithelial cells and mucus production, and in this case you see some eosinophilia and an impaired cytolytic response from IL-4 and 13 that leads to exaggerated and ineffective CD8 T cells. And the CD4 T cells, especially IL-13, can cause bronchoconstriction. So all these things probably contributed to what would have otherwise been less severe disease in this vaccine response. So in the lifespan of people, most severe disease happens in those very early months before 6 months of age. About half of the hospitalizations occur before 6 months, and the other half happen up through 5 years of age. And then there's another risk period in the elderly, especially the frail elderly, where there's a high rate of mortality in older individuals in many seasons, and when it's comparable to seasonal influenza. So RSV infects the ciliated bronchiolar epithelial cells, often sparing the basal epithelium, as you see here. And then the type 1 pneumocytes you're trying to compare this to what you know about COVID or SARS-CoV-2. SARS-CoV-2 infects these same ciliated bronchiolar epithelial cells, but infects the type 2 pneumocyte, the one that produces surfactant. And so there is some difference in disease pathogenesis, but a lot of the outcomes of obstructive airway disease is the same. It has a 15 kb genome. It's a pneumovirus. It used to be a paramyxovirus and was changed because of some of the gene order and in part because of what I'll show you in a few minutes. And we're going to focus on the F-glycoprotein today. It's one of 3 surface proteins of the virus. G is an attachment protein, and SH an ion channel similar to M2 in influenza. The F protein has been the target of most vaccine attempts over the years, and 3 phase 3 trials beginning in the late 80s, early 90s attempted to use F as a vaccine, either purified from virus or made in insect or mammalian cells, and it always ended up with about the same result, that you can boost neutralizing activity by 2 to 4-fold. And that was not enough to produce sufficient efficacy to meet the primary objectives of the studies. So in around 2008, Jason McClellan joined Peter Kwong's lab at the VRC. Peter led the structural biology group there, and Jason wanted to work on things other than HIV, and I really wanted to solve some structures in RSV, and Jason and I worked together to serially solve the structures of some of the epitopes for RSV, and then the post-fusion form, and then this pre-fusion form was captured when we found some antibodies that could hold the apical region together and then held the bottom together with fold-on trimerization domain. And you see where these antibodies bind. They bind the apex of the F protein in this pre-fusion form on a site that we call site 0. And compared to the antibody that's licensed for use in children born before 29 weeks gestation, palivizumab, these antibodies are 100 and even sometimes 1,000 times more potent than the antibody that is already licensed for use. So this is a new site of vulnerability on the virus that hadn't been seen before, and it doesn't exist after the protein rearranges. And so here's site 0 in red, and the rearranged form, you see it is lost, that surface is lost. So the post-fusion form, which was what all those other vaccines had been, doesn't express this epitope, which attracts very potent monoclonal antibodies. And note that this epitope is right in the hinge region where this protein has to unfold, and these alpha helical domains stack on each other to create the heptad repeat that ends up causing the fusion process. So through a series of protein engineering exercises, disulfides and cavity filling mutations were added to stabilize the prefusion in this state. And this was a collaboration between my lab and Peter's lab. And since then, it's allowed us to map the antigenic sites on RSV. There's now a new set of monoclonal antibodies that binds the stem region to kind of fill in, and it's going to be called site 6. But you see on the post-fusion form, there are some sites like site 4, especially in site 2, the palivizumab epitope in yellow. These 2 sites are on the post-fusion, and these are the only neutralizing determinants known prior to knowing about this pre-fusion structure. And that's why this molecule was felt to be adequate for vaccination, but just wasn't inducing sufficient potency of antibodies. Solving this structure also explained a few things that were not understood about RSV, and that is that looking at cryotomography, you could see a short and a long form of F, and this conforms perfectly to the pre and the post-fusion form. So F is flipping into the post-fusion state, even in the natural virus spontaneously. And we think this may have something to do with what happened during the vaccine enhanced disease. This vaccine was made by baking the virus for 72 hours at 36 degrees and then using a low concentration of formalin to presumably stabilize it. But when you do that with virus, almost all the virus is dead by 24 hours. And so we think what happened was that this heating process for 72 hours was killing the virus, but it was killing the virus by flipping all the F protein into the post-fusion state, therefore making it an inactive virus and a post-fusion F antigen. And we think that because we can do dot plots and show that the site 2, which is on both forms, was present all the way through this process. But antibodies that exclusively bind the pre-fusion form decayed at the same kinetics as the death of the virus. So we think this is what was happening, and whether that explains all of the vaccine-enhanced disease or not, that explains at least part of the vaccine character. And so we captured this structure, have a new site of vulnerability that doesn't exist on the post-fusion, and that protein boosted neutralizing activity 2 to 4-fold in several phase 3 trials. But now with this stabilized version of F, we can induce and boost neutralizing activity upwards of 10 to 20 fold. And the reason for this is that in the protein, if you match up the antigenic sites in this protein with the level of potency of antibodies, if we isolated several hundred antibodies from both infants and adults, and looking at those several hundred antibodies, the majority of them were to these top 2 antigenic sites that we call site 0 and site 5 that are not present in the post-fusion form. But note that there are some potent antibodies even on site 4 and some on site 3, which are present in both forms of the virus. And I'm going to show you something that we think about that. So this product was made at the VRC between about 2014 and 16. We did a phase 1 study doing a dose escalation with and without alum. And then because we were at the VRC with those PIs and Mario's group who does such beautiful flow cytometry, and now because we have these probe reagents, we could do a lot of work on the B cell response to the vaccine in people, in addition to just measuring antibody responses in serum. And so you can use the pre-fusion F on one axis and the post-fusion F on the other as probes and looking at B cell responses and be able to find antibodies that are B cells that exclusively bind the pre-F or that bind both fusion forms of the fusion protein or that exclusively bind the post-F version. We also have probes from Jason McClellan, who's now at UT Austin, for HMPV or human metapneumovirus, and I'll show you some data at the end about that. And so this not only allows us to look at individual cells post-immunization, but also look at activation markers and look at memory phenotypes of these cells over time. So if you look at this over time, in a pre-immune state, you have a few resting memory cells against pre-F, very few against dual-specific binders. And then shortly after vaccination, you get a big boost in the pre-fusion F exclusive B cells, the dual binding B cells. And over time, that diminishes, but even at a year, you see a fairly substantial preservation of new resting memory B cells that keep immunity up, we think, well past a year. And we were fortunate to have a collaboration with MedImmune because MedImmune did one of the last post-fusion F vaccine studies, and they also collected PBMCs. And so we can compare the response to the pre-fusion F, which does induce a lot of pre-fusion specific antibodies, and the post-fusion F, which is inducing antibodies, but they're almost all in this dual binding quadrant. And this will give you a much better response because most of the pre-F exclusive antibodies are much more potent in neutralization than dual binding antibodies, which are induced on an equal basis with the post-fusion F. Now, I don't have this slide in here today, but just so you'll know, what you're seeing here in these probes with the post-fusion on the bottom and the pre-fusion on the top are these strips or clusters of B cells. And so we also know and can map on an epitope level or an antigenic site level that these responses are to site 5, these are to site 0, these are to site 4 and 2 and 1. And so these clouds of B cells are actually B cells that are recognizing the same antigenic sites. And we have evidence for that as well. And so now you can actually map epitope-specific memory responses over time, because with these markers, you can see that the pre-F specific cells a week after vaccination are all in this activated memory quadrant. And you can show that they're mostly the pre-F exclusive sites are being primed by pre-fusion F, and post-F specific sites are being primed and activated by the post-fusion F, which is mostly this long 6-helix bundle, which attracts a lot of antibody response. And you can map this over time, looking at the phenotypes, and I'm just showing you here that same graph now, but only really categorized by these qualities. And so you can see this big activated memory response that eventually fades, and by week 44, it's almost all back in the resting memory B cell long-term memory pool. And so you can see why there is a big antibody response for a few months or after a vaccination like this, and then falls back to just a steady state with those memory cells. You can ask whether it's changed by one immunization or 2 immunization. We gave some people a 12-week boost, a 3-month boost, because we learned from other studies that at about this point in time, when most cells had gone back into the resting memory state, that's when people become boostable. And if they're primed, especially with a live virus or a vector, 12 to 16 weeks is when you can then be boosted to a high degree again. And you can see the B cell memory after 1 or 2 immunizations is not that different, except for this small hump here at 12 weeks. And if you look at the responses this way from 0 to 2, you see that they're about the same, and that there's no response at 12 to 14 in the 1-dose group, but there is some at the 2-dose group. But if you look at the very end, the 2-dose of immunization didn't really have a different set of memory B cell responses at 44 weeks than the single dose. So almost all the new vaccines are just using a single immunization, at least to get through that first season. Now, that site 3, the green one that I showed in the middle, there are antibodies that can bind site 3 in RSV, and also the site 3 in metapneumovirus. And one of the questions was whether the prefusion F would boost metapneumovirus specific neutralizing activity, or whether it could boost any kind of antibody binding to metapneumovirus. So with this probe, you can ask this question in the B cell compartment and show that antibodies or B cells that could respond to the prefusion F could make antibodies that also bound to the HMPV site 3 in that other virus. But that dual binding antibodies, which are mostly this cloud down here, don't boost that much of the cross-reactive type of HMPV. And you can show this here. This is antibodies that only bind HMPV, which stayed stable. And then there was cross-binding antibodies to HMPV in the vaccine immunized group. But unfortunately, these cross-binding antibodies are not contributing to neutralization. So this is not going to give you that much help against HMPV. There are many ways to stabilize the F protein, and now Pfizer has finally published their version. They used one of our cavity filling sites. They used a different disulfide site, and then they used a site at the base where these trimers come together down here just before the stem that Hans Langerdick had predicted would provide some stabilizing influence. The other thing that has been done is at this site where those helical domains stack to make the heptad repeat, if you introduce a proline there, as Hans Langerdick did several years ago, you can also largely stabilize this F protein. And just for those of you who've been working on COVID these last 3 years, if you take the S1 cap off of the COVID spike protein, what's left of S2 looks almost identical to this molecule here, or the prefusion F of RSV. There are other antibodies besides the ones that I showed you binding the top that can induce potent cross-neutralizing antibodies, and Merck is also developing an antibody that bound the apex that called B25 has now been turned into something called nirsevimab by AstraZeneca and Sanofi that will be used to treat babies at birth and keep them safe from severe RSV through about 6 months of age. Merck has this other antibody that is to site 4, but it's not a dual binding site 4 antibody. It is a quaternary antibody that goes across domains similar to this antibody AM14 that goes across from the site 4 side here to a site 5 hairpin, a beta hairpin that exists right here. And so this new antibody from Merck is kind of sold as a site 4, but it's really binding across multiple antigenic sites and is quaternary across proteins. So all this work was started back in around 2013. So 10 years later now, we're at this pivot point where all of a sudden we now have several products that have successfully gone through phase 3 trials. Some of them have BLAs accepted, and some of them are still either completed or just finishing phase 3. Some of them have already been through VRBPAC, and this one for the maternal vaccine from Pfizer will go through VRBPAC on Thursday. I think Helen is presenting some of that data. So we now have vaccines for the elderly based on proteins from GSK that was just recently approved a couple of weeks ago. That is a subtype A virus with an adjuvant. That's a half dose of what they give in the Shingrix vaccine. Pfizer has a subtype A and a B that they don't put with an adjuvant that also has a successful phase 3 result. And it should be coming up for review and hopefully approval by the FDA soon. Those two for the elderly, the maternal vaccine that we think will protect through also about 6 months of age, and mothers boosted and transferring antibody to their babies. And we'll see how that goes in a couple of days. There's also a vaccine from Janssen, the Ad26, given expressing the prefusion F in a membrane context, but also at the same time giving the prefusion F protein. That had a very successful phase 3 trial, and even maybe arguably better durability of response. But Janssen apparently after that has decided to drop their RSV programs for reasons that I don't fully understand. Moderna also has a vaccine that should be very close to being through phase 3 trials, and the Merck vaccine that'll be used like the AstraZeneca antibody. This antibody would be should be through phase 3 testing shortly. So we now have elderly antibodies that should get rid of that risk period, a maternal immunization that gets rid of a lot of this risk period, and now passive antibody that can be given directly to babies, even full-term babies, and get rid of this risk period. And we're still left with a period of risk for children between 6 and 60 months of age. And we don't know how severe that will be. We think that overall lung health will be dramatically improved by just protecting through 6 months of age when most lung development is completed. But there's still work to do on this age group. And because of the legacy of vaccine-enhanced disease, it's a more difficult regulatory process. And what we know can be safe are live viruses. And so live or live attenuated and recombinant or chimeric viruses are leading that effort. But we think that recombinant vectors or nucleic acid vaccines could also have low risk of causing the vaccine-enhanced disease based on what we know about the T cell response and if they present an antigen that it has the right structure. And then these other approaches will have a more difficult regulatory pathway. And so we have vaccines or passive antibody that can protect these different target populations for vaccine, and are still working on this young child age group. So solving the structure of F revealed this new target of vulnerability. We now have clinical proof of concept and structure-based vaccine design, and have subunit proteins, vector-based approaches, subunit proteins on nanoparticle display, and mRNA vaccines all coming along. And these vaccines have proven effective in phase 3. The finding that stabilizing class 1 fusion proteins to make them better vaccine antigens was the reason for starting the work and solving spike structures in coronavirus over that last 10 years, and was the basis for a lot of the COVID vaccines that came through. And we think it's a generalizable approach to improve vaccine antigens for these envelope viruses, and informs what we call a prototype pathogen approach for pandemic preparedness. And I'd like to thank my collaborators, Peter Kwong, Nan Chen did a lot of the serology assays, both in the preclinical and clinical studies, and Jason McClellan, of course, who is our long-term collaborator on structure. Our Chinese collaborators in Xiamen Medical University, Ningxia found the 5C4 antibody that was used to hold the top of the F together. And Michelle Crank and this group of people led by Tracy Ruckworth did a lot of the clinical analysis. And just thank my lab and the PIs who I miss from the vaccine research center. So I will stop there and take any questions you might have.
Great. Thank you so much, Barney. That was such a wonderful talk, and really sort of an overview of everything that's happened to bring us to where we are now with one vaccine licensed and several in the pipeline. So it is such an important time to hear about the history of how we got here. I'm going to go ahead and open it up for questions. Perhaps while we're waiting, I wanted to ask you, I think, one question, which is really around the work that you're doing about global equity and rollout. And I was just wondering what your thoughts were about the maternal vaccine / monoclonal antibody strategy for low-income settings. It seems as though it is possible that we will have both licensed for use in the United States. One is obviously easier to implement in low and middle income countries. And I'm just wondering what you think about what we will do when we move to sites outside of the United States, where it may not be possible to get monoclonal antibodies out, birth dose monoclonal antibodies, and whether or not there were some lessons from COVID that you think we could apply here.
Yes, thank you, Helen, for asking that question, because that's what I'm now trying to devote all my time to. And what happened during COVID was great, because we did get vaccines rapidly and we got them deployed in many places and it saved a lot of lives. But by the end of 2021, less than 5% of people on the African continent were vaccinated. And if you look at the analysis of deaths averted or mortality reduced and look over high and low income countries, almost all the averted deaths were in high and middle income countries, and relatively few averted deaths occurred in low income countries. And so there was a failure to really distribute and get the world vaccinated in 6 months, as I've said, instead of 6 years. And I think that could have blunted what Trevor described earlier, that these successive waves and allowing the virus to adapt and not getting the coronavirus under control fast enough is probably what's contributed a lot to the overall waves of variants that we now are experiencing. So it's in everybody's best interest to get this done more quickly. And RSV this work has been going on a long time, but there really still isn't a very good plan on how to get these products that work deployed into low and middle income countries, even though some of the studies for the efficacy trial were done in low and middle income countries, including South Africa and others in the Southern Hemisphere. And so the question is, how do we do this in a better way? And how do we get governments and companies and philanthropists and nonprofits and academics and everybody to agree that getting everyone vaccinated is a better approach for infectious diseases than only vaccinating subpopulations, because there's something like 120 million children born every year in the world, and only 4 million of those are in the US. And so we need to find ways of getting these products out. And of course, there is an advantage of having some of this discovered in the government, because government intellectual property is usually done by non-exclusive licensing instead of exclusive licensing. So the underlying IP for making prefusion F, and now there's several different ways that can be done, that IP is available non-exclusively, but the know-how for how to make the proteins or make the RNA or deliver things internationally is still lacking. And so just having the IP doesn't really solve the problem until you have the research infrastructure and facilities and know-how to make these things. And so a lot of my time is going to be spent trying to move better research capacities into Africa and eventually manufacturing into Africa. I think RNA gives us a chance to do that easier way, because it's relatively simpler than making protein bioreactors, and it's chemical synthesis. It can be done in small footprint, relatively small number of machines. And so anyway, that's what I'll be working on. And any advice or help you have about how to do that internationally is much appreciated. There is going to be a battle between maternal vaccination and passive antibody treatment of infants, because both are doing the same thing. And it's sort of the difference between 100 milligrams of antibody for the baby, or 100 micrograms of antibody of vaccine for the mother. And so the vaccine is probably going to be cheaper. And but there's cultural differences. Giving the antibody is probably a more secure way to get a particular level of neutralizing activity into serum. It doesn't require a healthy placenta necessarily, or a placenta that is transferring in the way that you want it to, and doesn't require an immune response from the mother. And so there's a lot of advantages of the antibody, but there's also advantages for the maternal immunization, because some cultures may not want you to touch their babies, and the cost and just the logistics of treating the baby versus getting to the mother. And I'm hoping some of the durability of this immune response will be sufficient that even in a postpartum immunization, you could protect a lot of future babies, because this is boosting to supernormal levels of neutralizing activity. And a lot of babies in developing countries, that if you have a first child, there's often a second child in about a third of the women within a couple of years. And so there's even a possibility for some postpartum strategies with vaccines. I'll try to give shorter answers.
That was a big question. So thank you. I'm going to open it up to others, and feel free to just ask the question rather than put it in the chat. Steve, did you want to go ahead and ask your question?
Yeah, okay. Hi, Steve Cunningham from Edinburgh in the UK. I really enjoyed that talk. It was fantastic. Thank you very much, Barney. You talked about the formalin inactivated platform and how that led to the issues, presumably with all of those vaccines. The GSK maternal vaccine study, of course, was stopped early recently because of an adverse effect with preterm births. The Pfizer maternal vaccine is the same technology, and so I just wondered, from your perspective, how likely are significant adverse effects related to the technology rather than the individual vaccines?
Right. Well, what is known in those older children and in adults, we know that you can safely give subunit vaccines because you're boosting pre-existing immune response. There's no, in my opinion, there's no risk of vaccine-enhanced disease from immunizing adults or people who've been previously infected, because all of that problem begins at the priming stage. So there were some reported preterm births in the GSK maternal vaccine, and so they've put that on pause and may give up on that. In my view of the data, the effects were pretty subtle, and there's no reported effects like that in the Merck and the Pfizer vaccine that I'm aware of. And so we'll hear the details of that on Thursday. But I think that, and Pfizer's giving just the protein, the two subtype A and B F proteins alone without adjuvant in both elderly and maternal. And so I think the risk for side effects in that population is very, very low. And the risk of passive antibody in the babies, whether it comes from the mother or from the monoclonal antibody, is very, very low. Palivizumab has been used since the late 90s.
Okay, thank you.
I was also going to say that a paper just came out a few days ago in Nature Medicine looking at individuals or babies who had received palivizumab and showing that they were able to generate a neutralizing antibody response to their first RSV infection. And that was one of the concerns with potential nirsevimab administration. So,
well, nirsevimab does bind a different part of the F protein. It binds the top, and so palivizumab binds the side. And so it doesn't really hide those apical epitopes. So, I think there's some studies that need to be done, but this happens in natural life anyway, because many mothers are transferring site 0 antibodies and babies eventually are able to fill in the gaps. But it does take several infections to get to a point at 5 years of age where you don't have as much disease. So, if nirsevimab inhibits or changes the pattern of how you fill in the gaps of your immunity to F or other parts of the virus, then I'm not very worried about that, but it could alter it to some extent how the repertoire is built over time.
Alpana, do you want to go ahead and ask your question?
Oh, sure. Yeah, sorry. Thank you so much, Dr. Graham. That was a fantastic talk. I was just curious if you had any thoughts about RSV prevention in immunocompromised hosts, especially given sort of the older children and other adults less than 65 that don't seem to be at least currently addressed in the ongoing and completed trials.
Right. Well, University of Washington, Seattle is the place most famous probably for studying bone marrow transplant, lung transplant, other immunocompromised people who have RSV or PIV3 infections that can be lethal in those populations, because we're talking about antibody, but obviously the CD8 T cell response is crucial for clearance of virus in those people, and that is really their major deficiency. And so people are at risk. Children in pediatric wards are at risk from nosocomial RSV, and adults with immunocompromised states, especially lung and allogenic bone marrow transplants, are at risk for nosocomial RSV or PIV3. And so there have been some studies to treat people with monoclonal antibody in those states that have been difficult. And I think it is because antibody is better for preventing infection and CD8 T cells are better for clearing the infection. And so whether antibodies will be used in those settings now to try to prevent RSV, I think is going to require additional study, but I think it's a good idea. I don't know that palivizumab was potent enough to do that, but nirsevimab is at least 10 times more potent than palivizumab. So I think it would have a good shot at prophylaxis in those adults. Still need something for PIV3.
Okay. Any other questions from the group?
Is it known what makes site 1 such a good antigenic site? Is it directly, are antibodies binding there directly block receptor binding?
Well, all of these antibodies work by fusion inhibition. The attachment for RSV happens either through this interesting circuitous route through the G glycoprotein, but a lot of RSV fusion, I think, happens spontaneously because there's a lot of polybasic groups and positive charge on the virus that attracts it to cells naturally. And when it gets close enough, the F can trigger and get it in. So these antibodies are not really preventing attachment, they're preventing fusion. And so they're fusion inhibiting antibodies. And the ones on top, might put some distance between those polybasic sites and the cell, but they also are directly binding those alpha helical coils that have to rearrange and stack. And so if you cover that up, you're covering up the trigger points for that rearrangement. And I don't know if that's the reason they're more potent or not, but they are definitely more potent. Most of them are more potent on a molecule per molecule basis.
Looks like Evan has his hand raised.
Hi, thanks for the talk, very helpful. Just wondering, I guess, has it been established what's driving some of the multi-season production that we're seeing from J&J and Pfizer? Just wondering if you had any thoughts there, and if this could be, maybe, if there's anything to understand the mechanism there. Thanks.
So could you say that again? Driving some of the what exactly?
The multi-season production that we saw, I think that you mentioned that we saw from J&J, and I think Pfizer described that they'd seen some protection through 2 seasons as well.
Right. Well, just as in COVID, people spend a lot of time looking at serum antibody, and serum antibody does wane over time, but even after a year, the level of neutralizing antibody in elderly serum is at least 4 times higher than it was before you started. And so you've still got quite a bit of antibody, and I think you've still got a lot more, maybe 10 times more memory B cells for F. And so, or in the case of coronavirus for spike. And I think if we spent more time measuring memory B cells and be less focused on serum antibodies, I think we would understand long-lasting immunity better. And I think RSV is people are so primed, you get reinfected with this all through your life. And so you're multiply primed, and this boost is likely, in my opinion, to last for more than a year. And RSV is kind of a settled virus. It's settled into 2 major subtypes, and it sort of toggles back and forth, and they co-circulate and find their niche, but they don't really drift. Every once in a while, there's new mutations that are introduced. Like there was a mutation in site 5 about 10 years ago that made the B subtype fail a Regeneron antibody that was focused right on that particular spot, but it doesn't drift that much. And so I think, I don't think an RSV vaccine boost in the elderly would have to be given every year. That would be my prediction. I don't know what is being planned. Need more data.
Yeah, I know GSK and Pfizer are both measuring out for several past 1 year, but I don't know that I've seen any of the data yet, past 12 months. I think we have time for just one final question. Jim, did you want to ask your question?
Oh, thanks, Helen. Thanks, Barney. Great talk. Wanted to ask, I saw a paper from you, Barney, a few years ago in a mouse model where you used like IgA and IgG, and you administered it intranasally, and you also did it parenterally. So I was just curious, what do you think is the best strategy for treating RSV now that we have good prevention strategies? Whether these antibodies are going to be potent enough, can we do it respiratory delivery or systemic delivery?
Well, I've never had much luck with respiratory delivery of antibody. I don't think it gets into the right place. I think it's probably swept away by the mucociliary blanket before it gets into that water space between the mucus and the top of the cell. And that's where the antibody needs to be, right at the top of the cell, right at where the cilia are living. And so most of the good effects I've seen from antibody, especially in the lower airway and lung, have come from systemically administered antibodies that transudate through the cell and get into that fluid underneath the mucus. And so my bias is that it's best to give antibodies parenterally. And if you're trying to prevent lung disease and lower airway disease with vaccines, it's probably best to immunize parenterally. And if you can, because the nasal immunization is unlikely to boost systemic antibody as much as a parenteral vaccine. These levels of antibody are very, very rare in naturally infected people. So these are way above what natural infection could elicit. And if you don't have high serum antibody, you may not protect against severe disease very well. And I've also not had much luck on treating post-infection with RSV or influenza. It was a big surprise to me, actually, that you could treat the people with coronavirus post-infection and still have a good outcome. That was surprising to me. And it was fortunate because it also means that the vaccine and anamnestic responses with the memory B cells are probably have time to actually do something and work. But yeah, treatment of RSV with antibody, maybe the new more potent antibodies would have a better effect, but treatment post-infection in animal models or in people that I've seen has not worked really well. And mucosally, even less.
Thanks, Barney.
Okay, all right. Well, thank you so much, Barney and Trevor, for speaking today. In the interest of time, I think we're going to go ahead and end this. As a reminder, this is recorded and we'll post this up on the website. And next time we have Professor Ann Dewar from HIV Vaccine Trials Network talking to us about development of HIV vaccines. So thank you so much to the speakers and for everyone for joining.